what did you do?
Hello.
Hello, who started the, can you turn off the broadcast? You should turn off the broadcast, Tanya. You should turn off the broadcast. Everybody is hearing.
I know, I didn't start it.
No, neither did I. I didn't touch anything.
Me neither. I was not on the computer in the moment.
Maybe Rosa did that then.
Yeah, probably she did by accident.
Can you stop the broadcast so that we can start it properly?
Maybe I'm trying.
Can you hear me?
But Francesco?
Yes.
I cannot stop the broadcast.
So to whoever is listening, we're having some trouble today. So the webinar has not started yet. So okay.
Hello.
Yes, Rosa.
Hello, how are you?
Good, but you must have hit some buttons, so you are already online and everybody's hearing what you're saying. Okay. So you probably hit some button here, and we are trying. I need to make you the presenter so that you can share your screen. Hold on a second.
Perhaps I used the bad link.
Something happened, but the broadcast is already started and everybody is hearing what we're saying. So
I can try to.
So be aware of this. Be aware of this. So now I made you the presenter and you can share the screen.
Okay.
Okay, so close. So whatever is on your computer, everybody can see. Okay. So be aware of the pop-ups of emails and that you may have. You know, everybody's hearing is okay. And now you can go full screen.
Okay.
Yeah.
Okay.
And what I suggest is that, so now Rosa is, it's 3:55. So I will wait until 4 o'clock.
Okay.
And then I will introduce you and give you a webinar, okay?
Okay, thank you, Francesco.
Okay. And so be aware that anything you say, everybody can hear it right now. You have a green microphone icon. If you want to mute your microphone, you should hit that one, okay?
Okay.
So I'll come back at 4 o'clock.
Okay, no problem.
So good afternoon, everybody, and welcome to this ERKNet webinar. My name is Francesco Emma, and I'm speaking from my office in Rome. As usual, before starting, let me remind you that your microphones are turned off, but that during and after the webinar, you can send your questions through the application that is present on your screen. I will ask your questions at the end of the presentation. The webinar will last approximately 30 minutes. We will then have 15 minutes available for questions. Our speaker today is Rosa Vargas-Poussou. Rosa is a pediatric nephrology with additional specialization in renal physiology and in genetics. Her main area of interest is hereditary renal tubular diseases. She's in charge of the molecular and genetic studies of tubular renal diseases at the genetic department of the European Georges Pompidou Hospital in Paris. She's also manager and coordinator of the French National Tubulopathies Network. The title of her talk today is Monogenic Causes of Hypertension. Please, Rosa.
Thank you, Francesco, for this kind introduction. So today's topic is monogenic forms of hypertension. We will discuss about forms related to a primary increase of sodium reabsorption, which increase of bulimia and are associated with low renin. We can classify these diseases taking into account several criteria. I will use kalemia and the involved nephron segment. In the first group, with hyperkalemia, we have an increase of sodium reabsorption in the distal convoluted tubule that corresponds to Gordon syndrome or pseudo-hypoaldosteronism type 2. In the group with hypokalemia, the increase in sodium reabsorption is located in the collecting duct, and we have three diseases: Liddle syndrome, due to a direct increase of INAC activity, the activation of mineralocorticoid receptor activity in Geller syndrome, or the active action of cortisol in apparent mineralocorticoid excess. We will discuss today the main characteristics of each one of these four diseases. We will begin with Gordon syndrome. This syndrome, also known as type 2 pseudo-hypoaldosteronism or familial hyperkalemic hypertension, was described by an Australian physician in the 1960s and is characterized by hypertension with normal renal function, hyperkalemia, moderate hyperchloremic acidosis, low renin, low normal or lower normal aldosterone, and high sensitivity to thiazide diuretics. In these two graphs, you can see that the degree of high blood pressure is mainly dependent on age, and there is also in here at the right a strong correlation between plasma potassium and plasma chloride. Most cases have an autosomal inheritance, as in this example of this family with hypertension in 3 generations, with typical clinical and biochemical characteristics and correction of hyperkalemia and high blood pressure with thiazide diuretics. Gordon syndrome is genetically heterogeneous. Linkage studies in the 1990s identify 5 different loci called A to E according to OMIM. In 2001, in 2001, genetic studies allowed the identification of Win4 and Win1 genes corresponding to loci B and C, and whole exome sequences allowed the identification of genes KLHL3 and KUL3 responsible for types D and E. The gene responsible for the type A is still unknown. The sensitivity to thiazide diuretics suggested an activation of the thiazide-sensitive sodium chloride cotransporter, or NCC. Nevertheless, the research of gain-of-function mutation was infructuous. The identification of Wing and 4 genes in families mutated in families with Gordon syndrome allowed the understanding of novel mechanisms implicated in the regulation of sodium reabsorption in the distal convoluted tubule. The first mutation detected in Wing1 here, this type of intronic deletion, increases the expression of the catalytic isoform or Wing1, L-Wing1. In contrast, mutations in Wing4 are point mutation specifically located in an acidic motif of Wing4. The 2 other genes code for Culin-3 and Kelch-like 3 proteins, which are components of an ubiquitin ligase complex and interact also with Wing kinases. Here, in this scheme of Kelch-3, you have the site of interaction with Culin-3 and the motif Kelch interact with kinase. Interestingly, the further studies showed the acidic motif of Wing1 interacts with Kelch3, and recently mutations in the acidic motif of Wing1 were showed also in patients with Gordon syndrome. Finally, in Culin-3 gene, mutations are always the splicing mutations that delete the exon 9 and the 57 corresponding amino acids, and mutation in these patients are often de novo. Mutations in one of these genes are responsible for the loss of interaction between these 4 proteins and an increase of Wing4 expression and upregulation of NCC with increase of sodium reabsorption and a reduction of potassium secretion with hyperkalemia. To explain the reduction of potassium secretion, there are several hypotheses. First, increased NCC activity reduces sodium delivery to ENaC decreasing electrogenic sodium reabsorption, and thus the drive for potassium secretion through KIR11 or ROMK channel. Nevertheless, this mechanism has not been confirmed in other conditions. The second hypothesis is a reduction in the mass of connecting duct segment, which has been observed in animal models, which could impair potassium secretion, but here again not confirmed in all models. And third hypothesis is a direct inhibitory effect of Wnt on potassium channels, but the mechanism is not clearly identified. If we look in more detail and summarize the current understanding, normally Culin-3 coordinates the enzymatic and substrate binding activities of the ubiquitin E3 ligase, and Cache3 is the substrate that recruits Wnt to the E3 ligase complex. After the ubiquitination process, Wnt are degraded by the proteasome. The loss of this interaction due to the mutation in one of these genes stops this process and increases the Wnt activity. We know that Wnt can be autophosphorylated, and here the role of the kidney-specific Wnt1 kinase is important for the phosphorylation, but also the low chloride intracellular concentration. Then they phosphorylate and activate two other kinases, SPAC and OSR1, which in turn phosphorylate NCC, increasing its activity. Wnt proteins modulate the function of other proteins that can explain other manifestations of Gordon syndrome. For example, hypercalciuria has been described mainly in patients with Wnt4 mutations. The mechanism is not clear, but two papers suggest a modulation of activity of calcium channels TRPV5 and 6 in the distal convoluted tubule or in the thick ascending limb. Concerning the acidosis, the classical view to explain it is that the reduction of sodium delivery to collecting duct decreases the electrogenic sodium reabsorption, and thus the drive for proton secretion, similar for potassium excretion. Nevertheless, it is known that other mechanisms of thiazide-sensitive sodium reabsorption takes place in the beta intercalated cells via the concerted action of pendrin here and the NDCB exchanger here. Studies in a model of Gordon syndrome with activation of Wnt4 showed an increase in beta intercalated cells and activation of this exchanger with increased sodium reabsorption and bicarbonate secretion. This last aspect can explain the metabolic acidosis observed in Gordon patients and can contribute to inhibition of potassium secretion. If we look now at the relationship between genotype and phenotype in a group of patients, we can observe that the Culin-3 patients have the more severe phenotype, significantly younger, higher hyperkalemia, and blood pressure for age, followed by recessive KH3 cases. In the other side, patients with mutations in the acidic motif of WING1 present the less severe phenotype with moderate hyperkalemia, with normal blood pressure or moderate hypertension. The more severe phenotype in patients with Culin-3 mutations is explained by an additional effect of Culin-3 on vascular tissue as shown in several animal models. In this recent work, the authors developed cell models that overexpress mutant Culin-3 and mouse models that express it ubiquitously or specifically in vascular smooth muscle cells. They provide evidence that Culin-3 related hypertension is caused by alteration in both renal and vascular functions. These animals have wall intima media thickness and impairment of vascular relaxation. This dual influence helps to explain the phenotype severity of patients with Culin-3 mutations. Finally, this slide summarizes in our experience the proportion of patients with each type, showing that KH3 related type 2V is the more frequent form, as you cannot also, the transmission could be autosomal dominant or autosomal recessive. But there are still about 1/3 of patients without genetic identification. We move on the second disease, which is apparent mineralocorticoid excess, or AME syndrome, which is a rare autosomal recessive disorder characterized by severe hypertension, low plasma renin and aldosterone, and hypokalemia. Affected patients display low birth weight and failure to thrive, polyuria and polydipsia, often nephrocalcinosis, and biochemically they have elevated urinary ratio of cortisol to cortisone metabolites. Some cases with mild phenotype have been described as type 2 AME. This syndrome is due to mutation in the HSD11B2 gene, resulting in a deficiency of the renal 11-beta HSD2 enzyme, which converts active cortisol in cortisone in the kidney. Normally, mineralocorticoid receptor has the same affinity for both aldosterone and cortisol. As the cortisol circulates at 100 to 1,000-fold higher levels than aldosterone, mineralocorticoid receptor is protected from cortisol activation by this enzyme. In the AME syndrome, the functioning of this enzyme is impaired, and therefore the mineralocorticoid receptor is occupied by cortisol, resulting in an increase of sodium reabsorption and potassium secretion. These two graphs illustrate the genotype-phenotype correlations in some fresh families. Mutants with residual function, residual enzyme activity in vitro, are associated with mild hypokalemia, mild hypertension, and lower ratio of tetrahydrocortisol to tetrahydrocortisone. A recent work of an international consortium has explored several aspects of this disease in a cohort of 36 patients. In the panel A, you can see the structure of the human gene showing non-mutation and those harbored by 36 patients in bold. And in panel B, the worldwide distribution of this cohort. Here you can see the high blood pressure, mean arterial blood pressure, the alkalosis, hypokalemia, and cortisol to cortisone metabolic ratios. The red open circle here represents a female with AMP type 2. The median age of the patient in this cohort was 4.6 years. The cohort was equally distributed concerning the sex. 86% of patients were born at 10 and 76% were small for gestational age. Interestingly, 75% of patients presented with nephrocalcinosis. This consortium developed a computational model of 11-beta HSD2 enzyme that provides a structural explanation for the different phenotypes. Globally, the mutations that cause severe AME are those enhancing dimerization and disrupting the binding between the enzyme and substrate or coenzyme. In the other side, mild phenotype disrupt indirectly the function of the enzyme. The follow-up in this cohort was available for 16 patients, and as you can see, the cardiovascular complications were prominent, with 3 patients dying from cardiac arrest at 16 and 70 years. A left ventricular hypertrophy present in 12 out of 16, hypertensive retinopathy in 10, and for the 13 surviving patients, 12 have persistent left ventricular hypertrophy, 3 aortic root dilation, and 2 aortic insufficiency. Other complications were observed, including nephrocalcinosis, and 4 patients developed chronic kidney disease after 10 years of diagnosis. 2 patients were received renal transplantation, and they observed a remission of the low renin hypertension and hypokalemia after the transplantation. We move now to the third disease, which is Liddle syndrome. This condition was first described by Liddle in 1963. It is transmitted also with an autosomal dominant pattern Its classical clinical and biochemical features are early onset hypertension, suppressed plasma renin activity, together with low plasma aldosterone, hypokalemia, and metabolic alkalosis. However, it can present with a milder clinical phenotype. This table describes the clinical characteristics of one mother and her 3 sons affected by the disease. You can see here the effect of anticipation with an early severe hypertension in affected sons compared with the mother. And you can also observe the response to amyloid treatment. This slide summarizes the main clinical and biological characteristics in a small group of patients diagnosed in our lab. You can see the hypertension, the low renin, mainly in supine position, the hypokalemia, sometimes normokalemia in a few patients, and the alkalosis. Liddle syndrome is due to gain of function mutation in one of the genes encoding for the 3 subunits of the epithelial sodium channel ENaC, here expressed in the principal cells of the collecting duct. ENaC is composed by 3 homologous subunits, alpha, beta, and gamma. Each subunit has 2 transmembrane domains and intracellular domains. Set termini in all ENaC subunits contain a proline-rich sequence or PY motif. Most of the mutations detected in this syndrome are frameshift, missense, or nonsense mutation, impairing the PY motif. Normally, PY motif is a binding site for NET4, which promotes the ubiquitination and internalization and proteasomal degradation of ENaC. Mutations in the PY motif of ENaC prevent this binding to NET4, resulting in accumulation of ENaC at the plasma membrane. Less frequently, the reported mutations led to the development of Liddle syndrome, increasing the sodium current and the opening of the channel without affecting channel density. It is the case of this recent report of one mutation in the extracellular domain of alpha ENaC, which causes Liddle syndrome by increasing intrinsic channel activity. The 4th and last disease is hypertension exacerbated by pregnancy or Geller syndrome. It is characterized by autosomal dominant transmission, an early onset of low renin and low aldosterone hypertension associated with hypokalemia, further worsening during pregnancy. This disease is extremely rare condition described only in one family so far. So in this family, a point missense mutation affecting the codon 810 of the NR3C2 gene encoding for the mineralocorticoid receptor was detected. Expression of this mutant demonstrated a significant degree of constitutive activation of this mutant that is also strongly activated by progesterone, explaining the exacerbation during pregnancy. Surprisingly, they observed also activation with spironolactone and with cortisone and 11-dehydrocortisosterone, explaining the hypertension observed in male patients and in non-pregnant women in this family. You can see here the clinical presentation and biological data of individuals of this family harboring or not the activating mutation, which early presentation of hypertension, low aldosterone, and metabolic alkalosis. This constitutive activation of mineralocorticoid receptor is, of course, responsible of activation of ENaC and sodium reabsorption and potassium secretion in the principal cells of the collecting duct. I would like to finish with a table summarizing the treatment of these 4 conditions. Of course, a low salt diet is recommended. For Gordon syndrome, patients are in generally adequately controlled by thiazide therapy. In severe forms and taking into account the vascular manifestation described, the amlodipine could be an option in patients with KUL3 mutations and severe hypertension. Patients affected by AME syndrome respond well to mineralocorticoid receptor antagonists, as the activation of this receptor is implicated in the pathogenesis of the disease, as well as to dexamethasone, which reduces the cortisol production. In Liddle syndrome, the specific treatment is represented by ENaC blockers, amiloride, or triamterene, associated with, of course, a low sodium diet. And for Liddle syndrome, the mineralocorticoid receptor antagonists obviously do not work because sodium reabsorption is independent from mineralocorticoid receptor activation. For hypertension exacerbated by pregnancy, amiloride is the preferred treatment, and obviously spironolactone is contraindicated. In conclusion, monogenic hypertension, primary in renal disease, include disease associated with increase in sodium reabsorption in the distal convoluted tubule and collecting duct. They are low renin hypertension with abnormal plasma potassium. For all of them, a precise diagnosis, including genetic diagnosis, is important for genetic counseling, specific and early treatment, and prevention of complications. Thank you for your attention, and I would like to remember you the next webinar.
Well, Rosa, thank you so much for this wonderful overview. I think that from your talk, I think that everybody now is well aware that you are one of the best experts in this field. And I think that really we learned a lot by listening to you today. To the audience, please send in your questions, and I will transmit them to Rosa. The first one comes from Dan Weering, and he is first of all, thanking you for this overview. And he says that it is known, he's asking whether it's known why 75% of patients with apparent mineralocorticoid excess suffer from nephrocalcinosis. And in fact, this was one of the questions that I also had for you. Can you illuminate us on this aspect?
The cause is not very well known. The authors that followed these cohorts attribute this nephrocalcinosis to hypokalemia and nephrointerstitial lesions secondary to hypokalemia with calcification after these fibrosis lesions. But there is not experimental and physiopathological evidence that this is the mechanism. Anyway, in this patient, the hypokalemia is important, is severe, and the polyuria and polydipsia, failure to thrive, and everything is also associated with this hypokalemia. And for the moment, I don't know more about the physiopathology of this nephrocalcinosis, but it's really very, very frequent.
Okay, thank you. And just to remain on the same subject, I was surprised by this adolescents that had a cardiac arrest and suffer from AMA. Was there an explanation on this? Do they have coronary disease?
I don't remember if there is a detail in the paper. I don't really know if they describe it a coronary disease. No, I don't know.
Okay.
I need to check in the paper, but I don't remember like that.
No. Now we have a question, and I hope I pronounced the name well, and if not, please forgive me, of Syed Saimul Hukwe. The question is why some monogenic hypertension causes polyuria and some not?
It should be related with in part with hypokalemia and with the age of presentation and other associated lesions. In particular, is frequent in AME because the patient have severe hypokalemia and nephrocalcinosis.
So this hypokalemia, just to be even more clear, does hypokalemia per se inhibits water reabsorption, or is it a consequence of tubular damage that also expressed in the form of nephrocalcinosis? Probably
both. It's known that severe hypokalemia inhibits aquaporin 2. But also that hypokalemia, severe hypokalemia could be responsible of Fanconi syndrome and sometimes of this type of cysts and interstitial fibrosis.
Okay, so I have a few more questions for you. In the meantime, please send in your questions. Sometimes, blocking the mineralocorticoid receptor has side effects. Do you think that amiloride for AMA is a bad choice, or do you still think that this could be a way of avoiding chronic inhibition with spironolactone?
Yes, in patients with secondary effects of mineralocorticoid antagonist, the amyloid could be a second choice for this for patients.
Okay, because just as a matter of discussion, because I would tend to consider this as actually a good treatment, because these patients probably need lifelong treatment, and
Yes.
this is why I was asking you this.
Yes.
We are receiving 2 mails, and again, I apologize if I don't pronounce well your name, from Naveen Kumar Matewada. The 2 questions are as follows. When should a patient be evaluated for monogenic causes of hypertension? And do you have any specific indications? I just read the 2 questions to you. So when do you think we should really consider that this could be genetic? Basically, this is the question.
The early presentation of hypertension is definitely a good indication to explore more the possibility to be genetic, and it's important to explore, to do a genealogic tree with information, with relevant information. And of course, if during the study of this patient, a young patient with hypertension, you find dyskalemia, hypokalemia or hyperkalemia, it's a good way to define families and patients that need a genetic evaluation.
So to summarize what you said, early onset, familiar cases, and when there are electrolyte abnormalities or acid-base abnormalities. Would this be, would you agree with this?
Yes.
Yes. So we're having a few more questions. Before that, let me ask you one question. You did not mention glucocorticoid remediable hypertension.
Yes, because now we consider that more in the group of primary hyperaldosteronism, which is more endocrine diseases.
Okay.
And because time is not it's not enough.
Okay. So just wanted to have your comment on that. So a few more questions from the same person that already asked you this question. The first one is Sietse Mulhukwe. Actually, 2 questions. Is there any drug which is absolutely contraindicated in monogenic hypertension? And sometimes we are bound to prescribe all forms of antihypertensive. If I interpret well this question, and please send me another email, another message if I'm not. Basically, what is asked to you is, are there any antihypertensive drug that in these conditions should not be used?
Well, I think that in most of patients with this disease, we don't need other hypotensive drugs, because in general, taking into account the mechanism, the diuretics specific of each segment are sufficient. As I said, for Culin 3 severe cases, probably the amlodipine could be an interesting option. In other cases, I don't really I don't have a lot of experience, but I think that there is no contraindication for other drugs. And I know that some patients with Liddle syndrome sometimes are more severe and could be could and yes, and need another drug.
Yes. So, but there basically are no clear contraindications. And I must say that, if I may, in my experience, through some of these cases of Liddle syndrome or AMA, sometimes the diuretic treatment is not enough, and sometimes you do need to add another drug. But what you're saying is that there is no contraindication. We have a question from Naveen Kumar Matewada, who asked the question before: during the treatment of these cases, do you target just the blood pressure, or will you try to correct the metabolic abnormalities also?
To me, the more important thing is blood pressure, but in general, the drugs corrected both. It's our experience. For example, for Gordon's syndrome, thiazides corrected hyperkalemia, and and the other is also the case with hypokalemia. But logically, the target should be the blood pressure.
Okay, thank you. Iman Nureldin asks you, first of all, thank you for a very interesting lecture, and ask you, what is the role of dexamethasone in the treatment of AMA? AME, sorry.
Yes, the role of dexamethasone is to reduce the cortisol production dependent on ACTH.
Okay, thank you. So, Gijs Franken is asking you, after thanking you for this overview, so that he imagines that kinases such as WINK1 and 4 are not only expressed within the kidney, and he was wondering whether a patient with mutation in WINK1 and 4 have symptoms that are not related to sodium reabsorption in the kidney.
I know that there are works with the kinase showing that kinases play a role in vessels, also in muscle cells sorry. But for the moment, the proof, the clear proof is in Culin-3 patients with mutation that there are evidence of a vascular effect, but probably because the defect in kinases is lethal when when there are homozygous knockout in and with cardiovascular important lesions, but the animals are not viable. So yes, there is effect, but by the moment the main effect is in kidney, and for Culin-3 we know that the effect in vessels is also important.
Okay, thank you. So if I understand well your answer, there might be a gene dosage effect whereby in a heterozygous state, the defect is primarily in the kidney, whereas if it's a homozygous, then you have a more broad defect that would not be compatible with life, right?
Yes.
And we have a last question, so I'm closing the question, and it's again from Seymour Ukwe. The question is it essential to keep the potassium above the normal level before treating hypertension? Would you like to try to answer this question? I suppose this is related to AME or loss syndrome.
The problem is that we treat the both things at the same time.
No. Yes,
So,
absolutely. So the treatment for hypertension is also the same treatment that will increase the potassium, right? So Rosa, again, thank you very, very much for this enlightening webinar. I think we all learned a lot, and maybe you can put the last slide again. And I invite you all to the next seminar, June 25, Dieter Hafner, XLH, X-linked hypophosphatemic rickets recommendation. Thank you to all of you, and goodbye. Thank you, Rosa.
thank you. Bye bye.